Aging begins at the cellular level. As researchers, I know you're all too familiar with the problem of senescent cells, those age cells that just won't quit. Not only do they lose their function, but they also act like tiny factories, pumping out inflammatory signals that make aging-related diseases, like Alzheimer's, arterial hardening, and diabetes, so much worse. But here's the kicker: to study or fight these problematic cells, I need a reliable way to identify them. Traditionally, that means tagging them with fluorescent or biochemical labels. It works, but it's messy, slow, and sometimes even changes the cell's own properties. Imagine wanting to study a cell's natural state, but accidentally altering it just to spot it. That's a real roadblock for research. Now, a team from Tokyo Metropolitan University has developed a solution that could change the entire field. Instead of chemical labels, they use electric fields to distinguish between young and aged cells, completely label-free. Here's how it works. By exposing cells to an alternating electric field, each cell rearranges its internal charges. The cell then moves back and forth, responding to changes in the field's frequency. The key is something called the cutoff frequency, the precise point where the cell's movement changes dramatically. That number, it turns out, is different for senescent cells compared to healthy, younger ones. Why? It comes down to the cell membrane. As cells age, the lipids in their membranes change, and this alters their electrical properties. By measuring how cells react to these frequencies, using a method called frequency-modulated dielectrophoresis, or FM-DEP, I can tell senescent from non-senescent cells in a matter of seconds, no labels required. The team focused on human dermal fibroblasts, cells critical in skin tissue. Their results? A clear, quantifiable difference between old and young cells, based on their cutoff frequencies. FM-DEP is fast, easy to implement, and crucially, doesn't interfere with the cell's natural state. This opens up a whole new toolkit, not just for basic aging research, but for regenerative medicine and drug screening as well. And because the method is label-free, I'm not limited to one cell type. The goal is to expand FM-DEP to other tissues, making it a versatile method for cell identification across disciplines. For medical researchers like us, this means faster experiments, purer results, and a clearer path toward understanding and maybe even treating the root causes of aging. If you want to dig deeper into the data, check out the work funded by JSPS Kakeni, grant numbers JP23 K28453 and JP23 KK0260. Thanks for watching. If you're excited by the possibilities of label-free cell testing, let me know in the comments how you'd use this in your own research.